In today's tricky topic video, we are going to look at cloning animals. We're going to look at cloning in plants and animals, and we're going to describe the reasons why cloning could be used in animals and plants. We're going to describe how plants can be cloned using cuttings or tissue culture. We're going to describe how animals can be cloned using embryo transplants and adult cell cloning. And we're going to evaluate the potential risk and ethical objections of cloning. At various points in this video, there will be opportunities for you to pause and complete questions, so make sure you have a pen and paper with you. So first of all, we need to identify what a clone is. A clone is an organism with the exact same nuclear DNA, so they are genetically identical. We're going to start the rest of this video by describing the reasons why cloning would be used in plants. So why do we clone plants? Humans have been cloning plants successfully for a very long time, over thousands of years. Let's look at the example of why humans might want to do this, looking at roses. If you wanted white roses, if you bred together two white flowers, you might expect to get white offspring. However, due to the complexities of inheritance, not all the offspring will necessarily be white. They might be pink or red due to recessive alleles. So, in order to get offspring with the exact characteristics we want, in this case white roses, cloning would guarantee those traits because the offspring would be genetically identical. Another example might be if a farmer wanted to guarantee the crops that they would produce would be free of disease. If they have one plant with disease resistance, they could clone those plants in order to have offspring that are also resistant. This has been used in lots of different commercial plants, such as bananas. Bananas are cloned in order to maintain characteristics such as the lack of seeds and also some disease resistance. Cloning is also incredibly important in plant conservation. Like animals, there are lots of plants that are critically endangered, and there are very small numbers in the wild. They are so rare because they don't reproduce very successfully in nature, and this is often because there's a lack of pollinators. Therefore, in order to keep this group of plants going, they can be cloned so that they do not become extinct. The example in the picture is a ghost orchid, which is critically endangered, but now can be cloned to produce new ghost orchids. One problem here that we're going to refer back to later is that it makes all the plants genetically identical. This is a problem because there's a lack of variation, so if a new disease comes along, this can take out all the plants. We're now going to go through step by step two different techniques for cloning plants. We're going to look at cuttings and we're going to look at tissue culture. So, how do we clone plants? We can clone plants because they have meristem tissue. This means they contain adult stem cells that can be used to create an entire plant. We can take cuttings or use tissue culture. To clone a plant by cutting is a very simple process that's been used for many thousands of years. There are 3 steps to this process. First of all, you cut a stem from the parent plant at a branching point because this is where there will be meristem tissue. Then you dip the cutting in a rooting powder that contains hormones that will encourage the meristem to differentiate into roots. You then plant the cutting into soil. So here is a pause point. Pause the video, see if you can recall those 3 steps, and then restart the video to check your answer. You can pause the video now. Okay, so you can now check your answers and see if you remembered those 3 steps. To make hundreds of cones, we can't use the cutting method very efficiently. So instead, we use the tissue culture method. This makes lots of genetically identical plants really quickly. This is a 4-step process. Step 1, a scientist would scrape off tiny bits of the plant which would just contain a few meristem cells from the parent plant. Then the tissue samples are placed in an agar, a sugar solution containing growth hormones, in this case auxin. The tissue samples then grow into tiny plantlets in the Petri dish, and the plantlets are then placed into compost and grown into plants. Take another pause point now, pause the video and see if you can remember those 4 steps. When you're ready, start the video again and you'll be able to self-assess your answers. You can now check your answers to see if you remembered the 4 steps. Now we're going to check your understanding using a pause point. For each question, you can pause the video, write down what you think the correct statement is, and then play the video again to check your answer. To make hundreds of clones, we would use tissue culture because cuttings would be too inefficient. At your second pause point, you can now pause the video again. So for this one, you should have got the offspring is genetically identical to its single parent. Offspring number 1 is incorrect because the environment can alter the phenotype, so it's not always identical. Number 3 is incorrect because although clones are likely to be non-identical to the parent due to the environment, we still say that they're genetically identical so that they are different to those reproduced through ordinary sexual reproduction. And 4 is incorrect because a clone comes from just one parent, not 2. You can now pause the slide and find the correct statement here. You should have realised that the correct order for cuttings is stem cut from the plant, cutting dipped in rooting powder, then it's planted in compost. Now see if you can select the correct statement to produce a clone by tissue culture. You can pause the video now. So for this one, you should have realised that step 1 is that the tissue sample is scraped from the plant. Step 2, this same tissue sample is placed in growth medium. Step 3, this grows into small plantlets. And step 4, the plantlets are planted into compost. We're now going to move on to looking at some of the risk and ethical objections to cloning plants. We looked at the example of the growth orchid earlier, and we're going to be able to evaluate the cloning of the orchid using an example exam question. You may pause the video now to read the question. One advantage of cloning is that we know that we can preserve this species, preventing extinction of the plant. However, a problem is that there is a low success rate, meaning we need to take lots of tissue which could damage the plant, and the clones will be genetically identical, which makes the gene pool smaller, meaning one disease could infect all the clones. You can pause the video again if you want to take time to read through this answer. You can now have a go at evaluating one example yourself. In this example, you're looking at the type of succulent, and you need to see if you can come up with 2 advantages, 2 disadvantages, and a conclusion. You can pause the video now to do this. For the next section, we are going to be looking at cloning in animals rather than plants. And while I describe the steps to do this, we are also going to make reference to some risks and some objections. So why would we want to clone animals? Like with plants, we want to know exactly what characteristics the offspring will sometimes have. This can be useful in agriculture, where we can clone animals with highly desirable characteristics, such as high milk production or high muscle production when farming meat. It's also useful, like in plants, for conservation. In 2001, this Asian gaur became the first animal to be cloned for conservation reasons. In this case, 692 eggs were taken from a common cow, which had their nuclei removed, and the nuclei of a male gaur inserted into the eggs. Another cow was then used to grow the fetus, and we had 3 successful embryos produced. This helped to continue the species. Unfortunately, the healthy born calf contracted an infection and later died, although this is said to have nothing to do with the cloning process. Another example, and one of many times in which the cloning process has been repeated, is the Pyrenean ibex, which went extinct in 2000. In 2003, scientists produced a live clone of the last ibex using a female domestic goat to carry the embryo. Unfortunately, the clone just died just minutes after birth due to defects. However, there is not only cases of failure in the example of cloning animals. Over 25 years, scientists from Japan have cloned mice and show that they have a normal, healthy lifespan. Research currently suggests that there is no reasons why a cloned organism would be less healthy than the animal it was cloned from. But it's important to bear in mind when you're writing evaluation essays on this that some people do worry about the risks of cloning for the health of the organisms involved. Commercial cloning, so for sale of bulls, has been taking place on a regular basis in the US and in South America. They want to do this in order to increase the number of high genetic merit animals, and they then sell the semen for commercial use, both in the countries of origin and also overseas. They want to sell the semen of bulls that have desirable traits, such as high levels of musculature. This process is not currently legal in the EU. We're now going to talk through step by step the processes for cloning animals, and there are 2 that we're going to look at. We're going to look at embryo transplants and animal cell cloning. So, cloning animals is actually much more difficult than plants because there aren't any adult stem cells like the meristem that we talked about in plants. No adult cells will be able to produce an entire organism using current technology, but there are 2 ways we can clone an organism. Embryo transplants are an artificial way to produce identical twins. Adult cell cloning, which has only been possible since 1958, with the cloning of a frog, is the other method that we are going to talk about. So, embryo transplants. First step is to collect a sperm and an egg from a male and a female. We then produce a fertilized egg. We then allow a zygote to grow by mitosis. This means that all the cells are the same in the zygote. We then divide the embryo into smaller groups of cells, and we implant those groups of cells into new surrogate mothers. These surrogate mothers can be the same species, but they can also be slightly different species. In this case, the embryos then grow into new organisms. The surrogate mothers will then give birth to organisms that are clones of each other. However, they will not be clones of either parent. This is an overall example diagram of what the process looks like. You can now pause the video and read through the process step by step. You are then going to start again and see if you can recall the process in 6 steps. See if you can lock this into your memory in 1 minute. Okay, that time should have been enough time for you to lock in the process. So pause the video again and see if you can write out all the 6 stages. You can now self-assess your answer using the stages on the screen. Play the video again when you are ready. We're now going to look at the process called adult cell cloning. In adult cell cloning, we first of all remove an egg from a female organism. We then remove the nucleus, so all the nuclear DNA is gone, and we end up with an enucleated egg. Next, we take the body cell of the organism that we want to clone. This could, for example, be a skin cell. We then remove the nucleus from that cell. We insert the nucleus from the organism we want to clone into the enucleated egg. We now have what would essentially be a zygote. We then give a shock to the egg containing the nuclear DNA from the organism we want to clone, and it stimulates the cells to divide. The embryo that is then produced is implanted into a surrogate mother. At this stage, it is also possible to split the embryo, as was done in the previous procedure, and implant multiple embryos to clone on a larger scale. The surrogate mother will eventually give birth to an organism that is the clone of the original organism that supplied the nuclear DNA. This is an overview of the whole process. You can now pause the video and read through the seven steps that we just went through, and then there will be an opportunity for you to recall these steps when you are ready. Okay, hopefully that time was enough time for you to learn it. See if you can now do this without looking back at any notes. Okay, you now may check what steps you wrote down and see if you were able to recall all 7 steps. Play the video again when you're ready to move on. We are now going to work through some quiz questions to check how well you understood this topic. For each question, pause the video, write down what you think the answer is, and then restart the video to self-assess your work. So in this case, animals are harder to clone than plants because animals don't have adult stem cells that can make a whole organism. They do, however, have some stem cells, which is why question 2 is not the correct answer, but these stem cells cannot create a whole organism. You can pause the video now and have a go at this question. So the correct answer is that we can make animal clones using embryo transplant and animal cell cloning. All the other answers involve techniques to clone plants. Pause the video now and see if you can identify the step mentioned. So the enucleated egg is demonstrated in step 1 because the nucleus is removed from the egg. Pause the video and see if you can identify the step in question. Step 3 was the ball of cells being implanted into the mother. We are now going to work through one exam question together. Pause the video and read through the question. Start the video again when you are ready to describe the cloning process. You can now pause this video and use these sentence starters to see if you can write out the step-by-step process for how to clone the source. Play the video again when you're ready to review your answer. Read through the full answer of steps and add into your notes anything that you might have missed. Play the video again when you're ready to move on. In this final question, you can pause the video, read through the stages, and see if you can independently describe from beginning to end the process of cloning Dolly the sheep, the first mammal to have been cloned by adult cell cloning. When you are ready, pause the video, write out your answer, and you can restart the video when you're ready to check your work. Here is a full explanation of how Dolly would have been cloned so that you can self-assess your answer. Pause the video now to take some time to review this. Okay, so in today's tricky topic video, we have gone through the reasons why we would clone animals and plants. We have described 2 processes to clone plants which is cuttings and tissue culture. We've described the two processes to clone animals, using embryo transplants and adult cell cloning, and we've evaluated the risks and potential objections to cloning. Thanks for listening.